Meiosis. So before the end of this video, you'll be able to do the following. You'll be able to describe the number of chromosomes in an organism as either diploid or haploid, understand the difference between male and female organisms, understands the difference between somatic and sex cells, and identify the source of genetic variability from meiosis, and be able to describe the general process of meiosis. So meiosis. Human cells contain 23 pairs of chromosomes. Each somatic cell or body cell contains 2 copies of each chromosome, which is 46 total. And each pair equals 2 copies of the same chromosome. They are homologous. Homologous just means the same type. One pair of chromosomes is called sex chromosomes, and in females we have 2 X chromosomes. In males, in humans, we have an X and a Y. The Y chromosome only contains the info that makes male a male, and the 2 X's give females all the information they need. Homologous pairs. They are similar in size and shape, they are similar in genetic info, and you get one from your mom and one from your dad. And there's genetic variability between the 2 of them, but they code for the same things. Crossing over, which is a vocabulary term, it occurs when homologous chromosomes exchange pieces of their chromatids during meiosis. Now remember, the chromatids is 1/2 of the chromosome. And meiosis is the process that we're going to be studying in just a moment. So, meiosis. Homologous chromosomes refers to chromosomes that have corresponding chromosome from the opposite sex parent. So you have one from mom, one from dad. And meiosis is the process of breaking down all of the genetic information into 4 haploid cells. So we're taking it from 1 cell, we're making 4. So we're going to go through 2 divisions. Alright, more vocabulary. Diploid, or 2N, is a cell containing 2 sets of 23 chromosomes, the somatic cells. Haploid is a cell containing only 1 set of chromosomes, or sex cells. These are going to only have 23. Our gametes are haploid. When we fuse these 2 gametes together to create a zygote, we have something that is diploid. Here's what you need to remember. Diploid, 2. Di, 2. 2 copies. We have 2 copies of everything. So this is where we're separating the X's. Haploid means that we're going to have 1 copy, so only 1 X. There's a difference between a gamete and a zygote. A gamete is going to be something that is haploid, and it's going to need to unite with another cell to form a new organism. A zygote is the cell that is formed by that union. So the sperm or egg cell is our gamete. The zygote would be the new organism that will be formed by the union of those 2 gametes. Fertilization happens when a sperm and an egg meet each other and they combine their genetic information. You get 23 chromosomes from mom and 23 chromosomes from dad. So we take 2 haploid cells and we make a diploid cell. So if both of these cells were diploid, if the egg from mom and the sperm from dad were diploid, then we would have too much genetic information, so it's necessary for us to cut that in half. So meiosis is only used for the production of sperm and eggs, or any of the gametes. It consists of 2 successive cell divisions producing 4 daughter cells. It has 2 main functions: reduce the number of chromosomes in the gamete to 23, and to ensure that every gamete is genetically unique. That's why we don't look exactly like our parents. We're an amalgamation or a combination of the 2. Meiosis goes through 2 divisions. We have meiosis 1 and meiosis 2. Meiosis 1, we have double the amount of genetic information, and meiosis 2 looks a lot like mitosis. In this, we have meiosis 1 where we replicate the DNA, we form 2 new cells. Then we have meiosis 2 and we have 4 new cells that we form. So double the amount of cells, reduce the amount of chromosomes by half, and end up with, from something diploid, something haploid. So interphase 1 is very similar to the mitosis interphase. Chromosomes will replicate in the S phase, just like in mitosis. Each duplicated chromosome will consist of 2 identical sister chromatids attached at their centromeres. So chromosome is the X, chromatids is the 1, and the centromere is what connects them. The centriole pairs will also replicate. In interphase 1, the nucleus and the nucleolus are visible, but they're going to start to break down. And this is what it's going to look like. So we have our chromatin, which would be the genetic material. We have our cell membrane around the cell. We have a nuclear membrane around the nucleus, and we have our nucleolus, which is the condensed chromatin. So meiosis 1 is going to have 4 different phases. Cell division that reduces the chromosome number by 1/2. So the 4 phases are going to be prophase 1, metaphase 1, anaphase 1, telophase 1. Those names seem very familiar. That's because they're the exact same names as mitosis, and it's very easy to remember them. So Now we're going through 2 divisions, and each one is going to have either a 1 or a 2 behind it. So homologous chromosomes are going to be chromosomes that are similar in shape and size. You get one from mom, one from dad, maternal and paternal. The homologous pairs or tetrads carry genes controlling the same inherited traits. So at the beginning of prophase 1, you're going to have 4 chromosomes that are going to pair up, okay, and they're going to form a tetrad. These tetrads have the same genes on them. So we have 23 pairs of homologous chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. So in prophase 1, we have synapsis, which is where the homologous chromosomes will meet up. Homologous chromosomes, they have the same genetic information. The sister chromatids are 1/2 of the chromosome. And the tetrad is going to be the 4 chromatids. So homologous chromosomes are going to have the same types of genetic information on them. You get one from mom, one from dad, and that's going to determine your eye color, your hair color, height, skin color, all those different things. So the sex chromosomes code for the sex of the offspring. If the offspring is going to have 2 X's, will be female. If it has 1 X, it's going to be a male. In humans, the sex chromosomes are the 23rd set. In a female, they're going to look very similar. They're going to look like X's. They're going to be the same height and everything. But in males, there's going to be one that looks like a normal X, but then the Y chromosome is much smaller. There's not nearly as much genetic information upon that chromosome. Still in prophase 1. Okay, still with me? We're still in prophase 1. So during prophase 1, when we have the tetrad that forms, we have those 4 chromatids together. Chromosomes are going to be doing a little dance. They're going to entwine. And this is where we're going to have crossing over happen. Crossing over is the source of all variation that will occur between non-sister chromatids at the chiasmata. During crossing over, segments of non-sister chromatids, okay, chromatids from different chromosomes, still homologous pairs but different chromosomes, break and reattach to the other chromatid. The chiasmata or the chiasma are the sites of the crossing over. A lot of C words that are going on here. So prophase 1 is the longest and most complex phase, and it spends 90% of its time in this phase. The chromosomes will condense. Synapsis occurs where the homologous chromosomes come together and form a tetrad. And we also have crossing over that will happen. So we have non-sister chromatids. The chiasmata is going to be the site of crossing over. They'll entwine and they'll switch information. So some of this information will end up in the new cells that are formed at the end of meiosis. So this is where some traits can be hidden for a generation and can show up because they will show up in different chromosomes because they have been moved around. And this is where a lot of genetic variation comes up in our population. Even though we're separating all these homologous chromosomes during meiosis, we're also creating different chromosomes in the process where we're sharing some of these bits and pieces of the information. You're not just getting mom's chromosome or dad's chromosome, you're getting a little bit of both. So it already gives us it multiplies the amount of different gametes we can get that is produced by the independent assortment. We have no control over which chromatid goes where, but by doing this we are also increasing the genetic variability, which allows us to sustain our populations. So prophase 1, we have our spindle fibers, we have our centromeres, our centrioles, we have our tetrad forming, we have crossing over that happens, we have all that good stuff. Then metaphase 1, shortest phase, they line up in the center, they align on the metaphase plate, independent assortment occurs, we don't know which way everything is going to go. The orientation is random, so during metaphase 1 it could be like this, or it could be the other way. And we don't really know how it's going to occur. So at anaphase 1, we separate the homologous chromosomes, so we separate the X's and they move towards the poles. The sister chromatids remain attached at their centromeres, so we still have our X's moving apart, and this is what it'll look like. Telophase 1, we now have a haploid set of chromosomes. We have one set, but we're going to divide it even further. Cytokinesis will occur and 2 haploid daughter cells are formed. So during telophase 1, this is what's going on, and we end up with 2 cells. Then meiosis 2 happens. We don't have an interphase 2. It's not going to duplicate any more DNA. If we do have a pause, it would be very, very short, but no more DNA replication. Meiosis 2 is very similar to mitosis in that we end up with the same product at the end where we have the one copy. Prophase 2 is the same as prophase and mitosis. We have the X's that are condensed. They attach to the spindle fibers through the centromere and all that good stuff. Then metaphase 2 is exactly the same as in mitosis. The X's line up on the metaphase plate, and we are connected at the centromeres. And then anaphase 2 is where the sister chromatids are going to separate. Remember, in anaphase 1 we separated the homologous chromosomes, the X's. Now we're separating the chromatids, the lines. Then telophase 2 is the same as telophase in mitosis. The nuclei reform, cytokinesis occurs, and we end up with 4 haploid daughter cells that are produced. These are going to be our gametes, our sperm or our egg. So this is what telophase 2 would look like. We have 4 genetically different haploid cells. So meiosis is going to be the process by which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. Haploid gamete cells are produced from these diploid 2N cells. So 4 gametes are made by cell division, by meiosis. The gamete cells have half the number of chromosomes as a cell. Now in males, as you'll see in this picture, we produce 4 healthy sperm. And men will produce sperm their entire adult lives. Women, however, are born with a set number of eggs at birth. And after we reach puberty, we start ejecting those particular eggs. But for every 1 egg that is formed, there's also 3 that are going to become polar bodies, meaning that they are going to be non-useful eggs, and they're not going to be ever used for sexual reproduction. So we have, for every 4 sperm that are produced, we only have 1 egg that is produced, which is standard in human females. We have 2 types of cell division here. We have asexual division and we have sexual division. Mitosis is asexual, meaning we're just making a complete genetically identical copy, But in meiosis, the biggest difference is that we're producing 4 genetically different haploid cells. They're genetically different. Now, the ones that are produced by mitosis, they are diploid. They have the 2 copies. But the ones that are produced by meiosis, they're haploid. They have half as much. In mitosis, we produce 2 cells. In meiosis, we produce 4. So mitosis is going to allow an organism's body to grow and replace its cells. It's used in asexual reproduction to produce a new organism, think prokaryotes, and a new daughter cell is identical to the parent cell. For meiosis, however, it is used in sexual reproduction to produce the gametes, and the new daughter cells are genetically different from the parent cells and from one another. So for every sperm or egg that is in the male or female, the genetic information that they contain is completely different. That's why, I have a little brother. Well, if all the cells were identically the same, then wouldn't I end up with another little sister that would be exactly like me? That's not how it works. That's why we have so much genetic variation within our populations. It's why we don't look exactly like our parents and our siblings don't look exactly like us. There is genetic variation due to the crossing over that happens at the beginning of prophase 1, where these homologous chromosomes, the tetrad forms, and we have the chiasmata that form and they cross over. Just to leave you with a funny little note, here's a fake ad for a single sperm cell seeking single egg to share a life together as a zygote and beyond. I hope you enjoy it, and don't forget to whisk.